So for one of our last videos for this section, we're going to be looking at probability. So we'd already talked about Mendel and kind of what he discovered and how he found that some traits are dominant and others are recessive, and that he had come up with a way to predict this. So we're going to be looking at one of the tools that's used to predict those possible outcomes. And that tool is called the Punnett square. So first of all, we need to assign some symbols to the alleles because writing out the alleles longhand is tedious. So we also have a system put in place to determine dominant from recessive. So any capital letters tell us the allele is the dominant version, and any lowercase letters tell us the alleles are recessive. Usually you like to choose letters that are somewhat different when you type them or write them out when it's capital versus uppercase. So using like A's is good because capital A looks different from lowercase. But C can be harder to distinguish because capital C and lowercase c can kind of look the same. It really does not necessarily matter what letter you choose as long as the letters for both alleles are the same, and the only difference between them is capital versus lowercase. Second, when we're doing our Punnett square, we have to determine what kind of gametes can the parents produce. Remember the laws of segregation from the previous video says that a gamete can only get one allele each. If you don't remember about the law of segregation, go back to the video before this looking at Mendel, and that can help you review that. So lastly, we need to set up our Punnett square. So we keep the letters the same when we're talking about the same gene, and usually we pick the first letter of the dominant trait. So let's say we're looking at red and pink. We would use R for red, capital R being red, lowercase r being pink. And then we distribute all the dominant alleles first. Now, remember that we can only have one letter for a single characteristic on a single gene. So let's go ahead and look at cross number 1, which we are crossing the true breeding parents. Remember, true breeding means that they can only make, they're homozygous, they can only make alleles or gametes that have that allele. So we're going to look at green leaves versus purple leaves. So the dominant trait is green, recessive is purple. And we're going to use capital G for green leaves and lowercase for purple leaves. And so if we look at the maternal genotype, we're homozygous dominant, and the paternal is homozygous recessive, which means our potential gametes can be capital G, or lowercase g or lowercase g. So we separate out and create the gametes. So we have maternal over the top and paternal over the side. When we fill that in, we end up with a Punnett square that looks like this. And so what we see is that every single square in our Punnett square has what we call heterozygous, one capital G and one lowercase g. So 100% of our genotype is heterozygous. And there's 0 for both of the other genotypes, both the homozygous dominant and homozygous recessive. The phenotype is 100% green. Since everyone's a heterozygous individual and the capital G green allele dominates over the recessive purple allele, everyone appears green and 0% appear purple. And so if we cross homozygous dominant with homozygous recessive, all of the offspring will be heterozygous and all of the offspring will have the dominant phenotype. But let's do a second cross, and we're going to take the offspring from the first cross and cross them together, which means we're crossing a heterozygote with another heterozygote. Both of these individuals have the potential of creating a capital G allele in the gametes or a lowercase allele in the gametes. And so we end up putting our cross together like this. And if we fill it in, we get a Punnett square that looks like this. Capital G, capital G, capital G lowercase, capital G lowercase g, and then lowercase lowercase. So we end up with one individual who is homozygous dominant, two individuals that are heterozygous, and one individual who's homozygous recessive. This gives us a 1 to 2 to 1 ratio for the genotype. Now, both the homozygous dominant and heterozygous individuals will show green as their phenotype. And the only individual who will have the purple phenotype is the homozygous recessive individual. So 75% of our potential offspring will be green and only 25% purple. So we end up with a 3 to 1 phenotypic ratio and a 1 to 2 to 1 genotypic ratio. That's a typo there. It should say genotypic ratio. Now, probability really does play a huge role in genetics, but it just helps us determine what the chances are. It doesn't tell us exactly, so we can't predict the future with like 100% certainty. We can just kind of tell us what are the options and what's the chance of each option happening. So if someone is homozygous, though, for a gene, we know that there's a 100% chance that the offspring will get that allele, because if you're homozygous, you can only give that one type of allele to your offspring. But if a person is heterozygous, it's a 50-50 shot of either allele. So if we look at what those different gametes could look like, right? If we look at this giraffe here who is albino, she's little m, so homozygous recessive. No matter what gamete we're looking at, it's going to have a little m because that's the only allele she can contribute, even with the laws of segregation in place. But with the male in this instance, he's heterozygous, which means there's a 50-50 chance that the sperm will be dominant for the allele, and there's a 50-50 chance it could be recessive for the allele. So there is a 50% chance that the offspring could be albino. Because what we do is we take the odds of the mom multiplied by the odds of the dad to give us the percentage chance of the offspring. Since mom is 100% albino for her gametes, we multiply by 1. Since it's a 50% chance of an albino gamete from dad, we multiply by 0.5. So 0.5 means it's a 50% chance. Now, if someone does show a dominant phenotype, how the heck do we know what genotype they even have? Because if there's a dominant phenotype, they could be homozygous dominant or heterozygous. So how are we supposed to tell the difference? One of the things we can use is a test cross. A test cross will take someone with an unknown dominant phenotype, so we don't know whether or not they're homozygous dominant or heterozygous, and cross them with a recessive phenotype and look at the offspring. Now, obviously, there are some ethical concerns when it comes to doing this with people. This is not something we would do with people, but we could do it with plants or small animals. So let's look at white alligators for our test cross. If we have the father, who he has pigment, but we don't know if he's homozygous dominant or heterozygous, and we cross him with a white homozygous mother, and we look at the offspring, we have two different possibilities. Either all of the offspring will be pigmented, or the offspring will be about a 50-50% difference, or 50-50% chance of being pigmented or white. So if the genotype of the father is homozygous dominant, then all of the offspring will be pigmented. If the male is heterozygous, then half of the offspring will be pigmented and half the offspring will be white. So basically, if you see any of the white offspring in this cross, that tells you that dad is heterozygous. But if all of the offspring have the same deep pigment, then dad has to be homozygous dominant. So let's do one of those test crosses together. So we have the green leaves and purple leaves again, and we don't necessarily know what is gonna happen with the father. So if we do if the father happens to be homozygous dominant, we will see these results, where all of the offspring have green leaves as heterozygotes. But if we do, let's say the parent is heterozygous, right? Our test cross comes from the homozygous recessive individual. We end up with a Punnett square that looks like this. And we see this breakdown instead, where half of the offspring are heterozygous with green leaves, and the other half are homozygous recessive with purple leaves. So if you ever see that 50-50 split, you know that the unknown dominant phenotype is a heterozygous individual.